very much forward to this talk on bacterial biofilms, phage therapy and chronic bacterial infections, the complexity, the efficacy and mystery. Thank you very much for coming.
The green light is for the green one.
This goes, that's the laser. How do you go about?
Yeah, it is in the computer. I'll try.
So I am not very good at speaking at a podium. I like to move around. This will be tough. Basically, this is something that I have been thinking about for a long time. I published a paper in January, and that paper was just some thoughts that I somehow had pulled together, I guess, in October of the previous year, last year. And I've been attempting to expand upon these ideas and also to think about how biofilms and chronic infections basically relate to one another and with phage therapy. And so I'm going to do this in slightly different order from what's shown up there. I'll start out with phage therapy. So I'm going to divide the talk into three parts. First of all, I'll be talking about phage therapy. Secondly, I'll be talking a little bit about biofilms, not a whole lot. Thirdly, I'll be talking about chronic bacterial infections and the idea of how we maybe should go about modeling them in experimental therapy of animals, model systems. And the biofilm stuff is based upon a number of publications that I have, and I'm thinking beyond that. So starting with the phage therapy stuff. So what is phage therapy? So it's approximately a 100-year-old use of bacterial viruses to combat especially pathogenic bacteria. When should we use phage therapy? We should use it when we have obvious antibiotic resistance. We should use it when we have antibiotic tolerance. So that means that we have bacteria that are not treatable with antibiotics, but when you take the bacteria out of the clinic and into the laboratory, they're still susceptible to the antibiotics. And then there's also the question of problems with the use of antibiotics, and increasingly we're understanding that antibiotics have detrimental impacts on our microbiomes. So this is the kind of crazy thing that I do. So this is doing Google Scholar searches on the phrase phage therapy, which is right there. And then these are just controls so that I make sure that I am understanding things correctly. And this right here is some kind of artifact. I think it's something to do with the Google algorithm. But you can see here that this is, I put this at 4 at the bottom. You can see here the use of phage therapy bounces along at a very low level. Probably because people were using a different word, or a lot of it was not published in English. They might have been using bacteriophage therapy, I wouldn't have called that. But you can see around the late 1990s, suddenly there's literally an exponential growth in the use of that phrase. And then we hit some kind of peak and it went down again. And now we've been going up again steadily since about 2003. And that's a doubling of about every 4 years in the use of phage therapy. So by that indicator, phage therapy is certainly growing. I gave this talk a couple of days, weeks ago, whatever, and people were covering their eyes. So this is my warning. So this comes from Betty Cutter, who is kind enough to allow me to show this. This is an article that is coming out, I believe, in July. And it is on the phage therapy of staphylococcus-infected diabetic ulcers. So these are toes. So what we've got here are people that had been going to their podiatrist. They were diabetic. They were being treated for these infections with antibiotics, and they couldn't cure the infections. And so these people had actually elected to have their toes cut off. And so instead, the doctor in this case had access to phages via Betty Cutter and treated these things with phages. And so this first case is a 4-week treatment with phages. I believe that they treated only once a week with these phages. They used debridement and everything to make sure they properly repaired the wounds. And this is over a 4-week period. And you can see you have basically healing. This is a person who wanted to have their toe cut off. Obviously, they didn't want to have it cut off. And this is another patient, and you can see this is 8 weeks of treatment. And it goes from basically a wound that's so bad they wanted to have their toe amputated instead to basically cured. And then what we have here is another case, and we have, at this point, pretty close to cured. I want to draw attention to the duration of the treatments. This was 18 weeks, so this is not happening in, say, 2 days. And I also want to make the point again that these are treatments that were not just one addition of the phages, one dosing, but instead multiple dosing over long periods of time. This I'm putting here because I read phage therapy papers, obviously, and I get frustrated, and so I just want to put the criteria that I have for reading it, what I want to know what's going on. I mean, there's all the characterization of the phages and whatever, but more than anything else, I want to know how many phages people are applying, and it's not always in the papers, or it's in the papers with MOI. We should call that the 3-letter word of phage therapy. So when you're putting just the ratio of phages to bacteria in these publications, first of all, what does that mean clinically? How are you going to possibly know how many bacteria are there? Because you can't know, therefore, the ratio of the phages to bacteria. The second problem is that then the reader has to sit down and figure out how many phages you added. I'll say this, that is so stupid. Don't do that. Please, put the titer. Titer is just the number of phages per ml. And if you're adding a certain volume, put that too. There's just, it's very frustrating. How often were the virions applied? Where and how were the virions applied, or the phages applied? What was the time delay? So this is for example, animal experiments. What was the time delay between the bacterial challenge and the virion addition? So I go straight to the materials and methods, and I look for the section where it's how was the experiment done? That's what I care about. And these are the things that I'm looking for. And then, of course, in the results section, what were the clinical, microbiological, and toxicological results? So from such information, we can then infer, though not necessarily in a straightforward manner, whether or not phage performance was adequate during phage therapy. But if you don't have this information, then all you have necessarily is whether it worked or not. Right now we're just starting to try to figure out how to understand what phage performance during phage therapy means. We really don't have, other than good results versus bad results, we don't have that good an idea. And figuring out what phage performance means during phage therapy, I'll get to what phage performance is in a second, probably means that we need better model systems for studying phage therapy, which is also something I'll be getting into. So what do I mean by phage performance? So I figure that there are 3 important things with regard to phages and phage therapy. And actually, since I come at phage therapy from an ecological perspective, these are the main points, I believe, the basis or the basics of what phage ecology is all about. They are the density of the phages, the density of the bacteria, and also what I call phage performance. The density of the phages, the more phages you have, the greater the impact it's going to be on the bacteria, mostly. With the bacteria, if you are requiring your bacteria to, your phages to replicate, then the more bacteria, the better, up to the point where there's so many there in stationary phase and they can't support phage replication. And what I mean by performance is how these phages are, performing. So things like their burst size and their Leica period, and their absorption rate, their ability to penetrate, which is an important word which I'm going to use. Host range, obviously, what organisms you can potentially treat. This is what I mean by phage performance. Oops, okay. So now I'm going to switch on to talking about biofilms, but I'll get back to this idea of performance, and I'll get back to phage therapy. So this is just a representation of a microcolony And all the complexity, the little oval things are bacteria. There's extracellular polymeric substances between the bacteria. There's other kinds of stuff. So basically, it's a bunch of bacteria that are sticking together. When an evolutionary biologist or evolutionary ecologist starts thinking about biofilms, they're immediately thinking about these clonal clumps of bacteria that form simply because the bacteria are replicating and not separating from one another. So that's the way I tend to think about these. Oops. So after years and years, I finally put together my own biofilm life cycle slide, and this is what it is. So you've got these cells that are, for whatever reason, moving about in different ways. They're able to attach to a surface. They can start replicating. They form these microcolonies, and then they might have some kind of dispersal stage. And I call this generalized because the way these biofilms do their things are going to differ between different bacteria. You're not going to have, for example, flagella on Staphylococcus. Or it might be that it's a more passive rather than active release of the dispersal bacteria. But that's not where I'm coming from. I actually got involved in thinking about the interaction of phages with biofilms by looking at phage plaques. So what we have here is an experiment which I've never really officially published, although it's in my 2011 book on phages and biofilms. What we have here is a T4R plaque, a rapid lysis plaque. This is the T4 wild type, and this is phage T7. The way I did this experiment, I just plated all three on one plate, and I counted on the fact that phage T7 was going to make huge plaques to have the T7 phage overwhelm the T4 phages. And as a consequence, maybe I'd be able to see the outline of a wild-type plaque. And what you can see here is indeed an outline of the wild-type plaque. The T7 phages are not penetrating into here, implying that what we have here are T4 infected cells, probably undergoing lysis inhibition. And if you measure the width of these plaques versus these plaques, they're about the same. I like to say that this is the last experiment I ever did. I looked in a microscope in 2005, and I looked at this, and I said, what are those dots? And I knew from reading Stent or Adams from the late 50s, early 60s, that bacterial lawns consist of microcolonies. And lo and behold, there were microcolonies. I could see it through the microscope. And from there, I realized that microcolonies might have some kind of impact on plaque growth, which I was interested in modeling plaque growth at the time. And from there, I said, wait a minute, biofilms also consist of microcolonies, and that's the genesis of my interest in phage interaction with biofilms. This past September, I think, I published a paper a very long paper, on comparing phages interacting with biofilms with antibiotics. And it's an ecological paper, and it's coming at it from the perspective of, would organisms that make antibiotics be using those antibiotics to disperse biofilms? Because of course antibiotics don't necessarily work as well as we want against biofilms. Versus phages and their potential to disperse biofilms. And what I came up with is a number of advantages to phages as used against biofilms versus the use of antibiotics. And you can see here, I mean, everybody talks about that some phages have these extracellular polymeric substances, depolymerases, and those are probably important, but it's certainly not the only thing. Phages use single hit kinetics. They only have to hit a bacteria with one phage to kill it. They concentrate their antibacterial activity in a very small area. One of those areas is actually inside of the infected cell, because they make more phages, they express genes. And also they generate new phages that don't necessarily diffuse very rapidly away from the site of release from infected bacteria. They're lytics. They blow these bacteria apart, maybe allowing the phages to penetrate further into the biofilms. They're bactericidal. Of course, not all antibiotics are bactericidal. They're evolved obligate predators of bacteria, and they increase their amplification in situ. Whereas antibiotics, I mean, they have some of these things, but they don't have all of them. And it's probably because of this that we can be effective using phages against biofilms in cases where we're not effective using antibiotics. And what I want to concentrate on, not that I'm going to be doing all that much concentrating on anything, is thinking about the penetration of phages into microcolonies or into biofilms, if you want to think about it that way. Up to this point, I've been thinking about the ecology of phage-biofilm interactions, really without thinking about their penetration into the biofilm itself. So the question is, that question mark there is showing penetration into the biofilm, and these are showing the movement of the phages along the surface of these microcolonies in order to infect and eradicate, penetrate into, propagate within the microcolony. And we really don't understand these fine-grained ecology of phage-biofilm interactions. So this basically, I'm thinking about the interaction of phages with mature biofilms. And I'm thinking about in terms of the interaction of the phage with one phage with one microcolony. And you can overwhelm the number of the biofilm microcolonies with phages, and you probably will see different results. So this is in comparison to not very mature, potentially more easily eradicated microcolonies and or the addition of larger numbers or many doses. So it might work differently if you have more than one phage. And what I am interested in is the phage's performance and their propagation through microcolonies, and otherwise their ability to eliminate mature microcolonies. And the short answer is that we don't have a great understanding of these things. So these ideas came about also from looking at plaques. I think it's in my 2008 paper with John in a chapter in my bacteriophage ecology book, where we were just kind of speculating on what is the origin of the cloudiness of plaques. And it probably has to do, of course, with there being bacteria there, but maybe it has to do with there being microcolonies there. And maybe what's happening is the bacteria microcolonies are maturing as the lawn matures, and they're becoming more difficult to eradicate by the phages, and thus you have an accumulation of cloudiness in these plaques. With the wild-type T4, you have lysis inhibition, so you have a real lack of ability to clear out these microcolonies. And all of these ideas kind of came together last fall when I had this thought. Actually, I'll put the next line up there. So maybe when we're looking at microcolonies, maybe when we're looking at biofilms, these biofilms, especially in the wild, can be both susceptible to phages and not susceptible at the same time. And the thinking was that when you have these microcolonies or biofilms, the outside of the structure are these cells that are relatively young physiologically and relatively easy to lyse. And they also may have immature extracellular polymeric substances. As you go further deeper into the biofilms or microcolonies, the cells get older. The EPS, in fact, is no longer as potentially penetratable. The idea is that you have a predator that's going for the young, juicy leaves on the plant, for example, rather than necessarily the more mature ones. And this is just speculation. The idea that older cells are in the middle and may be difficult for the phages to eliminate is certainly not mine. The idea that the EPS might be maturing in the course of the development of these microcolonies is mine. I mean, the idea that it might have an effect on phages is mine. And I want to just throw in the standard caveat: different combinations of phages and bacteria and circumstances are always going to be different. There's a lot of diversity out there. Okay, now I'm switching over to chronic bacterial infections. So this comes from Bruce Levin and Jim Bull, and they are not experts on chronic bacterial infections. They're microbial population biologists. But they have a nice quote, so I decided to use it. In chronic, this is from their 2004, I believe, Nature Reviews Microbiology Phage Thearpy article. In chronic infections, bacterial populations are maintained at substantial densities for extensive periods, years, or decades. Of course, Dr. Gorski alluded to. I put together a chapter for ASM this spring, and I tried to find all of the relatively recent human phage therapy papers, and these are they. There's actually not all that many of them. A large fraction of them are treatments of chronic infections. I also went through older papers, and I was struck by how treatment before we had antibiotics, you had an infection, you treated it with phages. You didn't wait 10 years before you treated with phages. So it is very likely that the treatment with phages that we're seeing today is more difficult because the infections have been around for much longer. So then I went to look at the literature on animal models of phage therapy, and I was interested in chronic infection, so I was wondering, well, what is the span of time between the bacterial challenge and the use of phages? And I came up with this table, so that's hours delay right there. This took me a long, very boring time to put together. And this is a sort of summary. The more than 1 day of bacterial growth before you add phages, we're talking about 10% of those studies. Naturally, if you're going to put that much time into putting together this data and organizing it, you're going to make a table. The table sort of shows a trend, but the trend really is meaningless. But what we are seeing is that people have been having a long delay between the bacterial challenge and treatment in these animal models, which is a good trend. But nonetheless, I asked myself, what exactly are these papers telling us? And so I went through all of the papers that had a longer than 24-hour delay between treatment and between the challenge and treatment. And this one is the latest one. It came out just this January, and it's a very interesting paper where they had rabbits and they had an osteomyelitis model, and they waited 2 weeks and 6 weeks before phage treatment, and then they treated with 4 different doses of phages. And although it's impressive they were able to sterilize the rabbits, it strikes me, and this reflects what Andre just said, that treatment success in this case seemed to be just too easy. I mean, it worked. It just simply worked. But in the real world, it doesn't simply work, as we know. So what would it take to have an animal model that really approximates what's going on clinically? And what properties might we hope such a model would have? So what is a model of a chronic infection? So what I thought of is that, well, you know, antibiotic tolerance is a big problem. We've got these chronic infections that may be susceptible to antibiotics in the laboratory, but not when you're actually treating. So why don't we use antibiotic tolerance, and especially the stabilization of antibiotic tolerance, as an indication that the infection, in fact, has stabilized. As antibiotics lose their ability to treat otherwise naive infections, as those infections progress, the biofilms are probably forming and maturing, and perhaps doing so in especially clinically meaningful manners. And such criteria may be particularly relevant if we're going to routinely wait until antibiotics fail before initiating phage therapy for treatment of antibiotic-tolerant infections. So what would be the ideal preclinical chronic infection, phage therapy infection? So first of all, obviously, multiple days and ideally multiple weeks between bacterial challenge and phage treatment. None of this, let's wait an hour and treat with the phages. Some demonstration of antibiotic tolerance, or at least showing that you've got mature biofilms in there. And then lastly, these models really should require multiple treatments, perhaps over days or weeks, in order for success. I mean, what we're trying to come up with is a way of understanding the treatment of chronic infections. We need a good model for that. So these are the kinds of things that we need to be able to see. So, if you want to hide your eyes. Yeah, I'm going to finish less than that. So, getting back to this. So we've got multiple weeks of treatment and multiple doses. And when we have antibiotic tolerance, we generally have these long-standing cultures or infections, and biofilms have formed, and it sets things up so we have things like persister cells, and we simply are not able to eliminate the infections using antibiotics. And it may very well be that the phage interactions with biofilms when we're treating chronic infections are very similar. The same things that give rise to antibiotic tolerance are probably giving rise to trouble in terms of your ability to eliminate these infections with phages. Not that you can't, but it takes time and it takes multiple doses.
Thank you so much. Thank you so much for this very clear and instructive talk and for keeping in time.
Thank you. I have no idea how much time that took.
So there are many questions I see. Could we get the microphone to the back?
Thank you. Roland Khazan from Hebrew University, Jerusalem. I wonder, why do you need to give multiple doses? I mean, if there is a resistance there, multiple doses won't help. And if it works, why then you need still to give more and more phages? I mean, I would guess that phages would grow and multiply within the microcolony. Why do you think you should give so many doses?
Yeah, so that's the assumption everybody makes, and that's really what I'm trying to argue against. It's not that the microcolonies or biofilms are resistant to the phages. It just takes time to penetrate those phages into the biofilms. A lot of times it takes surgery, so debridement, in order to have penetration.
But the phages are there. Why do you need to give more? I mean, you would expect that if they are sensitive, the phage will work slowly, but will work. Right?
So this gets back to the idea that these biofilms are probably phage susceptible and not phage susceptible at the same time. So in the real world outside of the clinic, in a pond or something, these biofilms probably are susceptible to phages, can sustain phage populations, but the phages are unlikely to be able to eliminate even necessarily the biofilms that they are infecting. We don't know this for sure, because we just don't have data. But what we do have is these chronic infections, and just one dose with the phages is not good enough to eliminate the infection. It just isn't. And probably what's going on is that the phages are able to get to these tender outer leaves of the biofilms, but they're just not good enough to penetrate all the way in, especially the more stationary phase interior of the microcolonies and biofilms. And so you need to hit them with the phages, lysis cells on the outside, hit them with phages again, lysis cells on the outside until you've eliminated the infection.
I would also suggest that maybe a high multiplicity, I mean, a lot of phages will also overcome resistant mechanism of the bacteria.
No, it's not a resistance mechanism. It's just
No,
an inability
mechanism.
it's an inability of the phages to support an ongoing replication by the phages in sufficient numbers. The phage performance is inadequate as they're penetrating into these microcolonies, and that's why you just have to hit them again and hit them again.
Thank you.
Okay, I think we are clearly behind the schedule and we have to move on. Sorry for that. Thank you again.